Francisco, one of the world's most beautiful cities. We are its master builders. Humanity, the product of over 100,000 years of evolution. The most capable, powerful life form on Earth. Or are we? Far tinier than the point of a sewing needle is another life form. A community of minute living things surrounds us, the product of almost 4 billion years of evolution. They greatly outnumber us, and their collective weight exceeds that of all other animals and plants combined. Microbes. The trillions of microscopic bacteria, viruses, protozoa, and fungi that are everywhere around and inside us. Most are beneficial to us. A few are not. All have amazing powers we are just beginning to recognize. This bacterium can survive 1,000 times the nuclear radiation that would kill a human. This virus is responsible for the colors of many tulips. Microbes like these generate half the oxygen we take in with every breath. The power of microbes is largely untapped. Now they promise new ways to fight disease, alleviate hunger, and clean up pollution. They promise to change our world. Microbes are commonly associated with illness. It's easy to forget that they are frontline warriors in the fight against disease. Moles are fungi that look like microscopic plants. This mold changed medical history.
Industrial monument to the miracle drug. Mass production penicillin plant at Terre Haute, Indiana, one of many where the life-saving medicine is now being manufactured wholesale. Tons of mold like that you've seen on spoiled foods are processed by rapid fermentation. The
Penicillin gave doctors a magic bullet. Suddenly they could cure people of infections that had claimed countless lives for centuries. Discovered in 1928 by Scottish microbiologist Alexander Fleming, penicillin was the first microbial product to cure human disease. The first mass-produced antibiotic. Like all living things, microbes compete with one another for what they need. Among their chemical weapons are antibiotics. They use them offensively and defensively. Microbes have had billions of years to perfect their chemical interactions. All that practice has made them the most brilliant chemists on Earth. When we discovered their powerful antibiotic creations, we became overconfident. We assumed these miracle drugs would serve us and our children indefinitely. We were wrong. Now there's a serious problem: antibiotic resistance. Around the world, we are losing our ability to cure infections. Vancouver, Canada. Julian Davies is a scientist, teacher, and entrepreneur. whose microbial research could save thousands of lives.
And most of the antibiotics that we have at the moment, we've been using for almost 50 years. And in 50 years' time, microbes have learned how to live with them. There are microbes that are found in hospitals that are causing severe infections in human beings that are resistant to every antibiotic that we know. They cannot be treated.
Tuberculosis, long considered under control in the developed world, is returning. So are many other illnesses we thought antibiotics could cure. Even though antibiotics work in different ways, microbes are outsmarting them one by one. Disease-causing bacteria colonize living tissue. Penicillin kills the bacteria by weakening their cell walls. When the walls collapse, the bacteria die. The patient's disease symptoms subside. Microbes reproduce by dividing in two, replicating themselves exactly each time. But mutations, the random genetic changes in all living things, have more impact among microbes because they reproduce so rapidly, as often as every 12 minutes. When a mutation enables a microbe to fend off an antibiotic attack, all the better for the new, stronger microbe, and all the worse for us. As antibiotics kill off the susceptible microbes, the resistant ones can multiply quickly without competition. So the more antibiotics we use, the more the playing field is cleared for antibiotic-resistant bacteria.
People ask for antibiotics when they shouldn't need antibiotics. Doctors prescribe antibiotics when they should not. As much to 40% of prescriptions written by family physicians are inappropriate. You don't give antibiotics for a cold.
Antibiotic resistance has reached crisis proportions. Around the world, thousands of people are dying of infections existing antibiotics can't touch.
We have to remember here that no matter what we do, microbes will become resistant to antibiotics. It is inevitable. What we can aim for all the time is to slow this process down.
Nothing will stop microbes from mutating. It's their nature to change.
So the question is, what are we going to do about this? And the obvious answer is that we need new antibiotics. The question then is, where do we find the new antibiotics?
The microbial community is everywhere. Largely unexplored, it contains many undiscovered antibiotics. So Davies searches close to home.
Oh, look, I found a feather. I can stick it in my hat, right? Yeah, sure. Yeah.
But great spot to take a soil sample.
Oh, by that tree? Oh, yeah. Let's have a look there. Right. So here I am, essentially in downtown Vancouver, Stanley Park, the center of Vancouver. This is not an exotic environment, although some people might think so. But here is some soil. This is a bag of soil we collected just down the road, right by an old Douglas fir stump that may have been cut down many years ago. The stump is old, but the soil is typical of that particular stump. That's what's interesting. And if we go somewhere else, next to another kind of tree, we find another kind of soil.
I don't think we need to worm.
No.
Now, why is it another kind of soil? Because the microbial population is totally different. And we would expect them to generate different antibiotics, a variety of different products.
Most of the microbes in Davies' soil samples can't be grown in the lab.
Thanks, Lulu.
Separated from their tiny communities, they die.
I wonder where would you think we should go from here? In a handful of soil, there are maybe as many as 5,000 to 10,000 different microbial species, and they live there as a community. They don't grow singly like they do in the lab. They're not growing on little petri plates with nutrients. They need the community in order to grow. In fact, 99.9% of the microbes from the environment cannot be grown in the lab. So the question is, how do we get at them?
Davies is using a new approach to find antibiotics from microbes that won't grow in the lab. He heads the University of British Columbia's microbiology department and is also the president of a new company on campus called Terrigen. Davies and his colleagues are removing the microbe's DNA, the genetic codes that contain all the instructions for the microorganism's internal processes.
We take the genetic material that we isolate from a soil or some non-cultivatable source of microbes, and we take that genetic information and we introduce it into what we call surrogate hosts. We introduce that information into other microbes that we can grow. We are manipulating DNA. We're taking DNA from one organism and we're putting it into another organism. And we're asking that the second organism, the recipient organism, can use that DNA to provide us with novel products.
Microbes are no strangers to accepting each other's DNA. In nature, they do it all the time.
Microbes have incredible abilities to exchange genetic information. So a microbe can be resistant to an antibiotic and transfer the ability to be resistant to that antibiotic to another microbe.
Sharing genes is a fact of life in the microbial world. Let's see that again. Tiny fragments of DNA, called plasmids, are transferred from one microbe to another. A single plasmid can pass on many different genetic traits, including antibiotic resistance. Terrigen takes small fragments of DNA, rather like plasmids, from microbes that can't be grown in the lab, and implants them in microbes that can. The genetically altered microbes are grown in cultures by a robot. Small paper discs are saturated with the chemicals the microbes produce. The discs are placed in colonies of harmful bacteria. Already it appears Terrigen has found an antibiotic.
Staphylococcus aureus is an important pathogen in hospital.
We've got this very nice zone of clearing where bacterial growth has been inhibited around this disc. So we know we've got an active antibiotic here.
The clear circle means the harmful bacteria are being killed by the antibiotic.
What's really exciting to us about this one now is that it's also showing activity against Staphylococcus aureus, one of the bacteria that's been causing a lot of problems in hospitals these days. Many of the strains that you find in hospitals are resistant to almost all drugs. We're going to carry on with the purification of this one, and we're really hoping that it might be Terrigen's first new antibiotic.
A promising start, but new antibiotics take years to reach the pharmacy shelf. The need is urgent, and the search is on around the world. Kiev, Ukraine, in the former Soviet Union. Berkeley, California microbiologist Jenny Hunter-Cervera has come here to find new antibiotics among some of the hardiest microbes on Earth. Ukraine's Institute of Microbiology appears unsophisticated by Western standards. Many of the methods employed here and much of the equipment are not leading edge. But the Institute's scientists are well trained in classical biology.
What this program offers is a chance to link two very different sciences, sort of the classics with the modern, and to actually have more knowledge in combining those two sciences.
Genetic engineering is beyond the capabilities of this laboratory. But there is a unique and forbidding source of genetically altered microbes nearby. Chernobyl's reactor number 4. On April 26, 1986, it exploded. A toxic plume of radioactivity spread across 100,000 square miles. It was a monumental catastrophe. At least 15,000 people have died as a result of the accident. Yuri Gleba is the director of the Ukrainian Institute and also works for an American company. He and Hunter Severa are on their way to the reactor complex. They are mounting a unique and potentially dangerous antibiotic safari in radioactive soil.
The whole Chernobyl area is a huge petri dish. Yes, it's a huge experimental dish where you have experiments going on at a very high rate.
There's a toxic effect from radiation where organisms will either be killed instantly because they can't deal with it, or some that have evolved over time will be able to handle it, but they too will now be changed in the way they function. And that's what we're going to be looking at, are these little changes that might end up in new chemicals that could be of value to antibiotics.
The Chernobyl nuclear plant is at the center of a heavily contaminated and completely evacuated circle, 18 miles in diameter.
We are about to enter this special zone. There are many other people who would like to enter, and they have to have special permit that says the purpose of the visit and gives them permission.
These evacuees have brought flowers for the graves of loved ones. Cancer-causing radioactive dust is still a danger near the reactor. Coveralls and masks will afford some protection.
There's always a little fear and anxiety, but that's good because it keeps you on your toes and aware that you'll follow all the safety guidelines.
What you see is city of Pripyat. It's an abandoned city. Total of 100,000 people have been evacuated from this area. They were forced to leave everything they have had here. It has been empty since that time. It has to stay empty for another 100 years. It's roughly 100, 120 microrad.
You could almost hear sounds of ghosts that once were there playing on the playground, serving dinner, going to bed. You can hear birds chirping, you can hear insects, you see new buds and shoots coming out, but yet you know life is gone forever as it was.
Reactor number 4. Thousands of workers encased it in a 12-story high concrete tomb. It has already begun to crack.
This is where so-called red forest was. Pine trees that have been killed because there was a flash of radioactivity 12 years ago. Now it's regrowing. It's really not worthwhile that you go further, okay?
For the first days after the accident, the winds blew to the northwest. Here, the radioactivity is most intense.
Unlike us humans or animals, plants can't walk, so they stay here. They have to cope with this situation, with this stressful ecological situation. Yeah, this is very unusual. Look at these needles here. They are much longer than normal. There were some physiological stresses that this plant went through, and maybe more than that, maybe some mutations that are causing this really very sick.
The microbes in the soil here have probably undergone similar mutations. They may be producing new antibiotics.
At Chernobyl, we have to be very quick when we collect the soil samples. There is that unknown fear as you're collecting the sample and watching the little Geiger counter go up in numbers. 5. Whoa, we got a 5. Oh my gosh, am I going to get any dose that might cause changes in me? We have to move fast so that we minimize the chance of exposure. So there is a little sort of anxiety. In one way, you're dying to know what's inside that soil sample that could be beneficial to man. And on the other hand, you're sitting there thinking, this could be very negative for me, and so let me get out of here. Yeah.
The Chernobyl samples now go to the Institute of Microbiology in Kiev, where the bacterial strains they contain will be cultured. Elena Kiprianova heads a research team there.
We have a big collection of strains, and we maintain these strains under the mineral oil. All our collection is kept in such tubes.
Even though most bacteria don't adapt well to life in test tubes, the Ukrainian scientists have successfully cultured more than 8,000 species from around the world.
These strains were isolated from the samples which were taken in Chernobyl reactor. And these bacteria are very interesting. Are they the same as the microorganisms which live in Kiev or in Ukraine, or they are some different because of the radiation? And did the radiation change their properties? Maybe there are some more biologically active microorganisms than the common bacteria which are living in the Ukrainian soils. Do you understand me?
Using traditional techniques Alexander Fleming might have employed, Chernobyl bacteria are cultivated in test tubes. The chemicals they leave behind in their nutrient solutions will be screened for new antibiotics in the United States.
Yuri and I have sort of the same philosophy that you only find what you look for in nature.
If you collect a sufficient number of sufficiently unusual organisms, you are going to find what you are looking for. It has been proven in science many, many times. So I don't see us as being programmed for failure at all. I'm sure we'll win. This trip to Chernobyl was a very emotional day for me. To try to recall the events and try to analyze the consequences. This is something that changed the history of many, many thousands, if not millions of people.
Had the winds blown to the south the day the Chernobyl reactor exploded, the 3 million citizens of Kiev, 80 miles away, might have had to abandon their city. They were lucky, but the tragedy continues to haunt this struggling country. Will the Chernobyl antibiotic search derive a benefit from the worst nuclear accident in history? Perhaps. With so many species of microbes yet to be studied, we will find new antibiotics, and they'll work for a while, until microbes change the rules of the game. Again. The village of Chunamina in Zimbabwe, southeastern Africa. There has been no rain for many weeks. The village's crops of corn and millet have failed. The only staple left is cassava, its starchy, edible tubers safe underground, protected from the scorching sun. Victor Masona is a Zimbabwean plant biologist.
Cassava is the only standing crop. When we dug up a few tubers, I mean, it was like everybody was going for them, even before they were cooked, which means there is evidence of hunger. Everyone is hungry.
Cassava has brought Masona to this village. The plant is eaten every day by 500 million people in Africa and around the world. Westerners seldom encounter it, except in tapioca pudding. But here, it is the difference between survival and starvation. When something goes wrong with this crop, there's nothing left to fall back on. And something is wrong.
So this is the African cassava mosaic virus. You know, it's a disease that affects cassava, and you can see it by the crinkling of the leaves. You know, there's a mosaic on the leaves. All the shoots that come out have got the same disease. So you can actually spread the disease by propagating cuttings from this plant. So it can also be spread by whiteflies, sucking on this plant, and then they go and suck on to the next plant, and that way they transmit the virus from one plant to another.
Masona's collaborator is plant biologist Ian Robertson of the University of Zimbabwe.
Throughout Africa, this material is dug out of the ground. They see these symptoms, but they don't know it's the virus. So they'll break off pieces and they'll pass them to their relatives. And they'll plant them, but they'll plant them with the virus in the stems. So when the thing grows, it'll be full of virus, even fuller than this. And the difference between this plant and this plant is this one will give you at least 30 tons per hectare, and this one will only give you 6 or 5 or 4 or 2.
The villain is the tiniest of all microbes, a virus. Viruses are parasites. They enter animal and plant cells and use their victims' biological machinery to reproduce. Viruses are difficult to fight. Pesticides could help defeat this virus by eradicating its insect carrier, the white fly. But chemical treatments are too expensive and also might contaminate the water supply. Victor Masona uses another line of attack, one that will genetically alter the cassava plants.
Our work involves the genetic transformation of cassava for virus resistance. What this means is that we are trying to introduce genes into cassava. We hope that at the end of the day we will have genetically transformed cassava which is resistant to African cassava mosaic virus.
If you get the gene in, then we can produce for them Victor's kind of stuff, which is going to be virus resistant. Then we begin to solve the problem. We have hopes within the next year or two to have virus resistant cassava for the whole of Africa, and that's 200 million peasants who depend on it for their livelihood. That's the reason why we need Victor to learn how to put a gene in, and that isn't easy.
Masona obtained a grant to pursue his experiments at the International Laboratory for Tropical Agricultural Biotechnology in La Jolla, California. He fights one microbe, the cassava mosaic virus, with another microbe called an agrobacterium. Agrobacteria routinely pass their genes into plant cells. In a test tube, Masona mixes a solution of agrobacteria and genes from the virus. An electric current punches holes in the agrobacteria. The mosaic virus genes enter. The agrobacteria then transfer the viral genes to cassava plant cells.
You're trying to vaccinate the plant, right? So we are putting a gene from the virus into the plant, so it becomes part of the plant.
Masona gives the plant just enough of the virus so it can defend itself when the real virus attacks.
And the agrobacterium transfers the gene to the cassava cells, and then we take the cells, culture them, and grow them into plants.
After 5 months of careful nurturing, Victor has a finished product, a generation of genetically altered cassava plants.
It's done properly.
The next step is to field test the transgenic plants in Zimbabwe's climate, with Zimbabwe's farmers, among Zimbabwe's whitefly population. Ian Robertson dreams of the day when Masona's plants will thrive throughout Africa.
I'll go through all this procedure of multiplying in the field, and in 3 years' time we'll have 200,000 for the next batch of people who need it. Then they'll get 30 tons per hectare every year, and their cousins will get it and their friends will get it, so suddenly we'll have 30 tons a year more or less permanently.
But it's not quite that simple. Genetic engineering is a powerful new technology. Its long-term effects are hard to predict. Zimbabwe's leaders are cautious. They're drafting regulations for the testing of transgenic plants. Masona cannot bring his transformed cassava into the country until the regulations are in place.
it can be very frustrating that the plants are sitting in my lab in California. I cannot proceed with the testing and the introduction of genetically engineered cassava if the regulations that guide these sort of introductions are not in place. So the people are hungry, but there's something that could help the situation, and yet I'm not able to bring it here because I'm not allowed to do so.
Biotech holds out the promise of enormous power. We can manipulate crops, animals, and even people. That's a dangerous power. So we need decent people to make the choices.
Scientists, like Masona, are sometimes accused of toying dangerously with Mother Nature, of trying to play God.
I don't think I'm playing God. I think if God would even be happy about that situation, because our aims are to help alleviate the problem of disease in cassava in this case, by obtaining crops that can be used by very desperate people. And I don't think that is bad. That plants or crops can also suffer from diseases. My research involves trying to put things into the crop, so that, or into the plant, so that it doesn't suffer from the disease.
Masona hopes he will soon be permitted to test his cassava plants in Zimbabwe. If they prove resistant to the cassava mosaic virus, a young biologist's manipulation of the power of microbes could nourish hundreds of millions of people.
Dicen que
the great age of machines has multiplied our powers. But a price has been paid. The federal government's 310 square mile Savannah River site in South Carolina is one of the most polluted tracts of land in the United States. People can't live here. Microbes can. They routinely adapt to contaminants that would sicken or kill us. Methanotrophs, bacteria found in soil nearly everywhere, survive by eating natural gas or methane. What if we could expand their diet? Terry Hazen is a specialist in bioremediation, the use of bacteria like methanotrophs to clean up contamination.
How can we take those microbes and really utilize their extreme abilities to biodegrade material and direct it towards specific pollutants that are found in the water, in the soil, and even in garbage?
Microbes survive by taking in nutrients and degrading them or breaking them down with powerful chemicals they manufacture called enzymes. In our own digestive tract, enzymes produced by microbes that live there help us digest our food. At the Savannah River site, radioactive materials for America's nuclear weapons were made for 40 years. This polluted the landscape. Microbes and their enzymes may have the power to clean it up.
When the Savannah River site was first built back in the early 50s, they started a process here where they used a lot of solvents. They stored that in those tanks, and then periodically they would release it through a pipe system underground, which was quite leaky. And so a lot of the contaminants leaked out into the ground along this linear path. And then it seeped down through the soil and got into the groundwater. Now that's where it's the problem, because once that material gets into the groundwater, then potentially it contaminates drinking water wells. These chlorinated solvents have been linked to leukemia in children and things like this.
Chlorinated solvents are among the most widespread and cleanup-resistant contaminants. Hazen tried something new. He harnessed the unique powers of methanotrophs by feeding them extra methane gas.
The bugs are great because they're everywhere, these methanotrophs in particular, and by adding the natural gas, they grow up to higher densities, higher concentrations, so they degrade the contaminants. Same enzyme that they use to degrade the methane and use the methane as a food will also degrade this contaminant. In fact, it'll degrade 250 other compounds.
The microbes turn the poisonous solvent into carbon dioxide and salt. Hazen inserted long pipes underground beneath the contamination and its natural population of methanotrophs. He pumped in air laced with methane gas. The better-fed methanotrophs population increased 10 billion times. In less than 2 years, their enzymes completely eliminated the contamination.
Well, what happens once we're done? Well, we just stop pumping in the methane, bacteria die back to their natural levels, and everything's back the way it was.
Given time and the right conditions, microbes can degrade almost anything. Hazen's approach simply accelerates natural processes. In a typical garbage dump, the most common waste is organic material, like household trash and paper. It degrades very slowly.
Everybody's heard the stories about being able to read newspapers that have been in a landfill for 50 years, okay? That's because there's not enough water there, there's not enough oxygen to get the biodegradation.
At this Georgia dump, Hazen and a local contractor are pumping air and water into the refuse. The result? The resident microbes thrive. They multiply rapidly. As their numbers increase, they degrade more trash faster.
It's so much better, such a better way to deal with our waste problems. I hope this spreads exponentially.
We can utilize resources generated doing this operation to close the loop on the solid waste issue and leave that waste generated during our generation stable, so that the next generation doesn't have to pick up that cost and add it to its operating cost.
Yeah, I think this is going to spread throughout the country. I am just super excited about this. What a tremendously active microbial environment this has become. It gets up over 140 degrees, 150 degrees Fahrenheit, and it's steamy. All of that is the microbes degrading so much material so fast that they're creating this heat.
Yeah, I was just looking at it. It's got a good smell, too. Yeah. Yeah, it got a funky smell. It's got a good cook smell.
You can actually use your senses to see how well this is working, because the smell is much lower than if we had fresh garbage. This has just sort of a musty odor, whereas over here, where they haven't been using this process, the smell is so overwhelming that it'll make you throw up.
With some extra air and water, microbes degrade the dump's organic waste in a fraction of the normal time.
So we're going from 30, 40 years or more, perhaps, down to just a couple of years, or maybe in some cases just a few months.
A faster cleanup also saves money. Hazen's microbial method benefits our budget, as well as the air, soil, and groundwater.
Okay, let's catch a big one. You know, here we are on the Savannah River, and 20 years ago, the river was so contaminated that you couldn't eat the fish here. Now, there's more fish, and they're cleaner than they have been in many, many years. You know, I'm really happy that I've been involved in that, and that I can actually see an impact on the environment ourselves. And did you see that one surface there? The big one just came up. You know, it's sort of interesting that the microbes, the microscopic organisms in this world, the smallest of God's creatures, actually have the greatest potential for curing the greatest problems.
Bioremediation techniques like Terry Hazen's are just beginning to catch on across the country and overseas. As we learn more about microbial appetites and how to stimulate them, we may turn microbes into our greatest allies in the war against pollution.
Marinos.
For over 200 years, we've been living in an age of machines, ushered in by the Industrial Revolution. Now, we are at the dawn of a new age.
This is the biological revolution. This is going to have a tremendous impact on our daily lives. I mean, we already know how microbes influence what we eat, how we dress, our health care, and now being able to understand or decipher their communication codes and understanding how they function with the structure of their communities. We're going to take that knowledge and even make better products for food, agriculture, enzymes, healthcare, and so forth.
The ultimate machinery is the machinery of life. We are beginning to understand its workings at the genetic level. Many of the coming changes will connect microbes with the power of electronics. At a new Silicon Valley company, a team of biologists and engineers is designing a machine that identifies microbes by their DNA fingerprints.
A prototype, which Bill had.
Kurt Peterson is the firm's president.
There is a revolution going on today in microbiology, and a lot of that revolution is coming from the fact that for many years we've been making integrated circuits, we've been making these little chips that process electronic signals, and we're starting to realize that those same techniques for making these electronic chips can be used to analyze microbes.
Peterson's instrument will capture DNA from harmful microbes that might be present in a sample of blood.
What you see behind me is the guts of Cepheid's instrument. It's a DNA capture chip. It's made with integrated circuit processing. And each one of those pillars is about 1/100th of the diameter of a human hair. This forest of thousands of pillars is designed to capture the DNA that might be flowing through in a solution such as blood. DNA from a microorganism gets captured, and we can extract that DNA and analyze it and determine what the microorganism is.
By identifying microbes in a sample of blood, the handheld device will diagnose illness in minutes. It will also detect harmful microbes in air, water, or food. Microelectronics are perhaps the supreme expression of the Industrial Revolution. Their marriage to the growing understanding of genetics will have profound repercussions.
I look at the potential of improving my children's life. New healthcare products, new foods, new ways to deal with cleaning up the environment. All this is going to happen in my lifetime, and it'll leave a legacy for my children to sort of build upon in the sense of their world being a better place thanks to the roles that microorganisms play in our daily lives.
The age of biology promises to make the Earth's tiniest creatures our most valued partners. Our collaboration with them has just begun. Yet they are already changing our lives. The promise is real. The challenge will be to meet it wisely.